Thanks very much for having me, guys. And I couldn't think of a better way to spend my birthday than give a paper, apparently. So yeah, thanks for accommodating me. I'm really excited to be here because as I hope to explicate today, I think there's some really productive terrain between the areas that you guys have been focusing on here and some of the work I've been doing in the laboratory contexts of the life sciences just across campus here. So I'll say a little bit about CBNS here, which is a centre of excellence that Matthew Kearns, Georgia Miller and I have been working with for the past few years. And what I'm really hoping to achieve today is to throw out some ideas for a book that Matthew and I have been working on, which is tentatively titled Moving Targets. And we're basically looking at the crisis of nanomedicine and drug development in the context of the emergence of precision medicine and this whole set of infrastructure around targeting that is attracting a lot of international attention. There's a lot of sort of biobanks being set up with the aim of exploiting different kinds of biomarkers and genes as particularly as drug targets. And I'm hoping to bring some of this work that we've been doing into dialogue with some of the work happening here. And this is all going to be very speculative, and I'm hoping to use it as a bit of a platform to figure out where we might collaborate down the track rather than presenting work that's fully formed, because this is far from that. So what I want to do is run through the kind of crisis that's animating the work in basic research across the campus here and in the other 14 or 15 centres that I've been collaborating with across Australia. There's a whole lot of these laboratories who are recipients of this Centre of Excellence funding. And so I just want to give a bit of background as to their relationship to drug development in the therapeutic space and some of the questions that are and problems that are animating that research. And then I'm really trying to get a few different theoretical frameworks into dialogue to make sense of this, and I'm hoping to sort of throw that out there so you can give me some feedback on whether this is really worth pursuing. And then I want to really sort of open out this question of valuation in the pharmaceutical economy in another couple of ways as well, thinking about changes in research funding and also the role of the human in all of this as well. So I'm going to run through some of this fairly quickly. So CBNS stands for the Centre for Convergent Bio-Nanoscience and Technology, and it's been described by one of our leaders as basically trying to bring predictability and standardisation to the empirical science of bio-nanomedicine. So practically what that involves is a whole lot of people putting nanoparticles into mice and then finding out what happens. So usually they're given some sort of cancer and they try to then reduce the size of that cancer. So these are some of the books that have been animating our thinking about what's been happening in these centres or in these laboratories. We've got about 400 colleagues who are chemists and biologists, and they're really working at this interface between these animal models and developing new molecular polymers, basically. So what we've been really trying to understand is how all of these particular developments are animating the work of our colleagues. So some of you may know this concept of the patent cliff. So in the pharmaceutical sector, the whole model that's been animating the development of drugs has been, or new therapies, has been this concept of the blockbuster, which is once global sales reach over $1 billion, it's considered a blockbuster drug, and the profits from that then subsidise R&D in other sectors, as well as financing all of the payoffs to shareholders and the rest of it. So I'll explain a little bit more of that in a second. There's also all these issues around an ageing population and changes to welfare spending and spending priorities. So there was a big OECD review of this in 2016 of really how expenditures in reimbursements in the pharmaceutical sector have changed. And there's a lot of really interesting findings out of that report that, that the spend on orphan drugs is massively increasing. So an orphan drug in Australia is defined as a drug with that's directed to a disease that occurs in less than 1 in 2,000 of the population, or in the US, it's a total population of 200,000 that is the potential market for the drug. And with orphan drugs, they've gone from about 15% of total expenditure in reimbursements in OECD countries, and they're projected to increase to 20% in the non-generic space by 2020. So it's just becoming this bigger and bigger share of reimbursements by Medicare through the PBS and through other national systems. So we're trying to make sense of quality-adjusted life years and these econometric qualifications in the assessment of these drugs. And the initial reading, to sort of cut to the chase of that part of the abstract of the talk, is that they've been incredibly limited in their impact on policy making. What we're seeing is a sort of consolidation of policy making amongst ministerial powers that are really favouring certain cohorts over others with very little regard to these more utilitarian forms of calculation. And one of the other really interesting things of that OECD report is that they also flag that high prices generally are stopping people from accessing drugs. So they in particular flag the incredibly high cost of new hepatitis C drugs in Poland, which is also attached, of course, to various forms of stigma, and that this is also a massive issue in anti-cancer drugs. So among 12 new anti-cancer drugs approved by the FDA in 2012, only one actually provided survival gains more than 2 months. So there's this sense that this whole kind of blockbuster model, which I'll explain in a moment a little bit more about, is really faltering. And there's really very kind of incremental gains in the existing drug development pipeline. There's also this absolute proliferation in diagnoses and forms of categories of illness that is only increasing as biobanks get larger, as genomic screening technologies proliferate and get cheaper, and as new forms of mutation and illness proliferate as well. So this is the sort of public-facing image of the US's All of Us program, which is one of the largest in national databases, which aims to really increase this capacity to diagnose new forms of illness and ultimately find new forms of disease against which therapeutic drugs could be developed. So this is the kind of drop-off in pharmaceutical R&D spending by corporations that we've been sort of dwelling with, and which animates some of our colleagues' concerns and thinking. What we have here is, on the right-hand side, the total amount of R&D spending in pharma, and then this is the sort of growth rate, and it just absolutely plummets with the global financial crisis. And then this is just to sort of reiterate this point that the price per life year gained of new oncology medicines is massively increasing. And the sense amongst oncologists and people who have done empirical research around this is that they're actually deeply skeptical about the point of developing new cancer medicines, or whether they're actually worth what is being asked of them, basically. So this is a version of the blockbuster model that you see very often put forward, that you have sort of 10,000 compounds as a potential source of new drugs that the more promising ones of these, and I'll say a little bit more about what a promising compound looks like in a moment, are then moved into the sort of preclinical phases. They're tested out on animal models, and then eventually they reach human trials, and this is the kind of the famed sort of valley of death, where the clinical trials get larger, and the forms of evidence required of these drugs basically changes and increases in each setting in different ways. So I've basically been doing work at this end of the spectrum with people who are both developing new chemicals and are sort of moving towards preclinical stage. And one of the really defining features of the laboratories I've been working with is that there's no sort of clean separation in their minds or even in their physical laboratories between these areas. That you have people literally selling compounds for testing in amongst their development of new compounds. They have FDA-approved drugs like doxorubicin that they're testing on animals. So there's really sort of no sense that the laboratory exists in one part of this model. So this is a picture of a DOX particle, or a nanoparticle, that has been pegylated, as they say. It's had a number of compounds added to it, which are kind of like bits of Velcro that are aimed to basically there's a hope that these will stick to the right receptor on the body, and that the drugs will basically then be released once this part of the compound reaches. So this is just a little video showing how my colleagues have been trying to communicate this to the outside world. It's a very sort of sci-fi atmosphere where this drug has found a particular receptor, that it's being taken up, and that it then enters the cell, and then it sort of releases its cargo once it's in there. So we're sort of having discussions with our colleagues up in art and design about what a more realistic representation of this would look like, rather than something that's quite kind of ethereal and quite beautiful. If you're thinking about literally billions of particles in this kind of dark space, and they're sort of bouncing around everywhere, what might that actually look like to represent? So we've been sort of chatting to them a little bit about that as sort of the next phase of the project, because within the field there is a sense that this model isn't working, up to the point of someone doing a systematic review showing that for the last 10 years, only 0.07% of new nanomedicines, so this is sort of refined next step doxorubicins, are actually hitting their targets. So this is conceived really as a problem of translation about not just what's happening in the laboratory, but how new drugs are being developed from this analysis. So this paper was published by a Canadian group who I just had the fortune to visit in Toronto, and there's a whole sort of set of social dynamics that we are documenting in the book about how and why this piece was written and the kind of reception it had in the field that I won't go into today. But this sort of crisis in the drug development pipeline is very much animating the field. So one of their responses is to ask for more data from their colleagues and to delve into this kind of dark data issue, that people aren't reporting negative results, that they aren't reporting the same sort of categories in their research, and that journals are one way to start changing this issue. So there's this whole discussion around the sort of minimum reporting standards and what that might look like for the field and how to get people along. Another sort of set of responses to this crisis is to basically extend this kind of imagination into schools and into various publics, and sort of get them interested in, you know, the wonder of science. So this is a high school group which has visited my colleagues up at UQ, and these are basically virtual mice models that they can sort of play with in a VR system. And they can see how and where these anti-cancer drugs are accumulating in the mouse. So what's going on here? So one way to think about this set of developments that is occurring both in the pharmaceutical economy and amongst my laboratory colleagues is to think about a kind of reallocation of the places of judgment. This is something I'm trying to make sense of, is how and why and where judgments actually take place. So in the pragmatic sociology of Luc Boltanski and Thévenot, they talk about these kinds of critical situations that we encounter on a day-to-day basis and in modern institutions that kind of interrupt ordinary and stable relations. And this is about confronting uncertainty. It's about this kind of temporality of saying we need to figure out a way to move forward and come to some sort of agreement. So there hasn't really been much use of pragmatic sociology and this sort of theorization in the lab studies tradition, just because there is so much happening in these laboratories. It's very hard to just say, oh, there's some sort of industrial justification here, or there's some sort of critical moment where there's a recourse to the common good, just because the sites of the laboratory are so dispersed and so incredibly sort of seething with all of this activity. So in their analysis and in their book on justification, you know, Boltanski talks about the market justifications, industrial justifications, creativity as these kinds of orders of worth, this kind of grammar of how we make sense of valuation. And one of the things they really underscore is the need for this kind of critical knowledge to actually interrupt the flow of information. So I just sort of want to set this up as a kind of theoretical way to think about some of the problems of valuation. And what I'm trying to do is bring this body of literature in dialogue with what Andrew Barry has called informational enrichment, which comes out of his ethnographic work in the laboratory, where he argues that materials are qualified in laboratories, and they're qualified in all sorts of ways, in the fact that competition, marketing, regulation, IP, standards, they're all there in the laboratory. There's no outside where these things exist in the laboratory. So this lovely quote from Stengers and Benson's History of Chemistry, this material that's qualified in the laboratory then comes to act not in controlled laboratory settings, but in living labyrinths whose topology varies in time, where partial and circumstantial causalities are so intertwined that they escape any a priori intelligibility. So this idea of informational enrichment and these kinds of forms of critical knowledge, I think, are what I'm sort of toying with here conceptually. And then one of the other ways that this has been taken up in the literature, this wonderful review piece by Anita Harden in one of the anthropology review journals, where she really builds on Andrew Barry's work and sort of charts the different ways this concept of informational enrichment has been taken up in STS anthropology and sociology of medicine. And there's sort of this overlay of these of the kind of Anthropocene, but really it's sort of separating out this more traditional lab studies tradition, which is where we would really situate our work, this kind of state market corporate nexus as another sort of site in which people have been understanding information flows and understanding the qualification of pharmaceuticals. And under this, we could think about the sort of informal economies in which people are trading drugs, We could think about arbitrage in counterfeits. There's a whole sort of body of literature that people like Lindsay McGoey have been working on. We could also think about issues of trade and regulation and the fact that sanctions stop certain drugs from reaching people that they might otherwise reach. And I think, yeah, this sort of state-corporate nexus is really one of the other parts that we're working with in the book. But they also document quite beautifully what they talk about, the kind of re-scripting and the downstream informing of pharmaceuticals. We could also think of the ways that clinical settings actually are crucial to the way that the effects of drugs become known and they're sort of qualified in different settings. And this is all sort of bread and butter for you guys, these latter two categories. And thinking about people like Anne-Marie Moll's work and Kane's work on care and the kind of actualization and de-actualization of the effects of drugs in different settings. And then in this review, they sort of overlay this fifth category of environment, toxicity, causation, epigenetics, and gene regulation as ways in which both the laboratory is being configured and reconfigured, and the certainty of knowledge in the laboratory is being called into question. So people like Michelle Murphy and Hannah Landecker's work would be central to this. So this is, I guess, a sort of invitation to think about how we could, speak across these different sorts of fields, and some of the work I've been doing around doxorubicin and anti-cancer drugs is sort of moving down to this space here. Now, these two forms of drugs might seem fairly arbitrary choices, and I'm no expert on the former category there, on the former drug at all, but my colleagues have been doing a lot of work with doxorubicin. I've seen mice injected with it on several occasions, and it's really bread and butter for the models that they're working with in terms of developing new anti-cancer medicines. So I sort of got thinking about how we might think about these two wildly divergent categories of medicines, an opioid and an anti-cancer agent, and what commonalities they have. And we could say that they're both products of European industrial modernity in some way, and they're both indebted to receptor theory for their formation. They're both listed on WHO's essential medicines, but they have wildly divergent imagined subjects, and I think that's sort of part of the appeal of throwing these two forms of drugs together and trying to understand how the kind of informational flow around them and their forms of qualification are different. In terms of receptor theory, a big part of our book is documenting its emergence and this lineage between the work of Paul Ehrlich in the late 19th century and the development of nanomedicines. This is really building on the work of Bernadette Benson-Vincent, who's sort of charted this lineage and the role of sort of ballistic metaphors more generally in nanomedicine development. Ehrlich was actually the subject of a biopic, which is quite an incredible piece of film from the 1940s, which was all about this kind of supposedly salacious story of the development of this, magic bullet of the sort of originary drug of whose name escapes me right now. So one of the tools I'm working with to try and understand the ways that we could think about the flow of information and the understanding of drugs is to look at the way terms coexist in different fields and the way they're clustered around drugs and their development. So this is some computational analysis of the first 2 decades of dox research. This is 1972 to 92. And what I've done is just extract this from the Web of Science. This is the top 20% of cited articles. And what we can see is from the outset, the way the scientific fields or the way publication around this drug was really emerged is in two very distinct clusters. One of them around drug development and the other one around clinical trials. And this is, terms like advanced breast cancer, patient survival rate, partial response, and over here we have concepts of resistance, mouse models, and further upstream in the lab process. So as will become clear, what's really notable about this is sort of the absences of certain subjects as much as what's present. This shows the development of DOCS research and the most recent 500 papers. And this is a co-citation, or rather a co-term analysis amongst the abstracts and titles. So what this shows is basically the kind of epistemic communities talking about doxorubicin over the past few years. And what we see is that it's very much tied to imaging and the promise of new medicines in the ways that my colleagues across the campus are talking about. But these two kinds of clusters are still dominant here. And then we're seeing the emergence of polyethylene glycol, this PEG, as the kind of key development on the lab side. That they're basically trying to add all of these different polymers to the outside of the DOCS particle and find new ways to get it into tumours, essentially. This is just a tool called VosViewer, which is an open source Java program. It's quite easy to use, and it, I think, is a really interesting way to understand how scientific fields develop over time. What we see when we contrast this with methadone is basically a sense that HIV and the drug user are there at its very emergence. So we have the subject there from the beginning in the scientific literature. And this is the first 40 years, and a similar number of citations as with DOCS. It's, I think, really interesting to see how this sort of analysis pulls out certain subjects, that we have man and woman and drug user as distinctive within the scientific literature, alongside the more scientific concepts, or supposedly more scientific concepts of, morphine, methadone, and pharmacokinetics. And then what we see when we look at analysis over the last decade is really the proliferation of different kinds of subjects in this space. So, as well as the sort of consolidation of the abuse literature and misuse literature here, there's all sorts of ways in which gender arrives, that we have all these other sorts of qualifications, this kind of evidence-based medicine and Cochrane analysis style cluster up the top here sort of arrives on the scene, and that we can see quite a different set of dynamics. But yeah, so these are just some tools I'm playing with to sort of make sense of the way in which drugs are sort of qualified in both the laboratory and different experimental settings outside the laboratory in sort of policy experiments as well, I guess. So one of the other ways of thinking about these sort of crises in the pharmaceutical pipeline that we're also developing in the book and in another paper is to look at, is to really sort of broaden out the analysis of who are the subjects of these medicines. So one of our colleagues up in Queensland is not just using rats and mice, but also doing some experiments with dogs, which might seem like a sort of strange detour to the uninitiated. But there's really a sort of open question as to whether this is a patient of these new drugs or whether it's a kind of animal model in the same vein as the mice. And I was really reminded of, this kind of imagery of the first dog in space when I first saw this image. Like you've got this sort of modernist frontier narrative playing out in this photo here. But this is sort of no joke in the pharmaceutical economy, right? That animals are actually really key recipients of therapies and of drugs particularly. And we could think about pain medication in this context as well. That there are 24 billion chickens on the planet, that there's 1 billion cattle. Hannah Landick has done some great work on arsenic as a growth promoter, which emerged in the sort of 20s and 30s. And there's also the issue around domestic pets and companion animals, that 2/3 of Australians in the US have a household pet, and that pet foods are kind of a site of regulation and concern. And also that this sort of precision medicine discourse doesn't just stop with humans, that this development of biobanks is happening with things like the dog genomic database as well, and that dogs actually share a lot of our illnesses like dementia and diabetes, obesity, OCD, allergies, cataracts, and of course cancer. That's a whole chapter of the book looking at this stuff. I guess it's a way of opening up the question of really what's at stake in this issue of valuation. That there's this question of who are the models and who are the patients of all of this infrastructure and funding. And so one of the other ways that we're opening this out is looking at federal health research and expenditure. This is all very schematic. I'm going to try and tie this up fairly quickly and open it out for questions. But what we're really noticing is a kind of seismic shift in the sort of locus of power of decision making around health research in the country. And it's really notable that we're seeing, this sort of flatlining of community health and public health in comparison to the enormous growth in biomedical research. One of the papers that Matt and I are working on at the moment is also around this Medical Research Future Fund, which some of you might be aware of.
The fuss is for heart disease.
The single biggest killer of Australians.
The fuss is for dementia.
And there's thousands diagnosed every month.
The fuss is for mental health.
For the diseases we know nothing about.
The fuss is that without investment, medical research in Australia will fall behind.
And hope for ourselves and our loved ones
will disappear.
So the fuss is for all of us.
And for our future.
Yeah, so it's an incredibly succinct summary of some of the issues that we're grappling with in this space. That this is absolutely central to the biomedical research economy and to the pharmaceutical economy. Thank you, autoplay. There's this kind of almost explicit economy of hope that's being invoked here, but as we're kind of unpacking in this other part of the book, this economy of hope was introduced in the context of really savage cuts to public health, and the GP co-payment was also sort of floated alongside it. And what we're seeing is basically it's a very speculative genomics health mission and all these other sorts of funding sources. I won't go into where the other money is going explicitly, but this is a really basic breakdown that most of it is basically going into prop up falling pharma R&D in the form of explicit translation funding, propping up clinical trials funding, and then there's about $78 million going to NHMRC fellows, and then just a very small amount to help out with primary care research, and the $125 million mental health mission. So to just sort of try and wrap this up a little bit, the sort of concepts that we're really trying to make sense of here, I'm trying to sort of make sense of how really I was sort of dwelling on it this morning, and I think one of the ways to sort of filter this is, does precision medicine actually produce facts? Where are the facts in the biomedical economy now? Because there's sort of two ways we could think about this. One of them would be to say that, following Hannah Landecker and this sort of Anthropocene line, that actually, that dashboards and all of the kind of nudging that is going on in the health space is actually displacing more of the forms of disciplinary power that institutions like hospitals and universities are used to exercising, and that this kind of capacity for critical judgment is being displaced altogether. If a sort of counterpoint to that would be to say, look at tumour boards, look at the sort of ongoing power that universities have, that the hospitals still have hierarchies in the same sorts of ways. So maybe this isn't necessarily the case. So another way to think about this might be to say that in Deleuze's terms, that societies of control are actually sort of parasitically dependent upon societies of discipline, and that if you look at the way the political developments like robo-debt and the kinds of welfare shaming, as well as the way privacy concerns about the My Health record have been dealt with, that we have a much more complicated relationship between these forms of power arising here. So this is a really sort of big picture, sort of schematic way that I'm toying with to try and make sense of how and why critical judgment seems so difficult to arrive at in the laboratories, and to make sense of this, the kind of changes in the pharmaceutical economy. But I guess the sort of more normative point to end on is that lab studies folks have really emphasised the kind of singularity of the experiment in all of this, that we need to sort of attend to the different sites in which scientists work on their own terms, and to sort of understand their topologies in different ways. That there's a broader question about whose lives we're being measured against in all of this. The development of all of this precision medicine infrastructure and all these ways in which we're being sort of nudged and tweaked by Fitbits and all the rest of it. And there's sort of that really overriding normative question is, how can we actually live better together with each other's differences in this context? So thanks very much. And these are my collaborators, who I would like to acknowledge, but take full responsibility for all the errors in the aforementioned. Thank you very much.
Thank you. Okay, so we've got quite a few minutes to have some questions. I will remind you to please speak into this non-projecting microphone so the audio can be captured. Has anyone got a question they'd like to ask Declan? Don't be put off by the microphone. It's okay. It's not that hard to talk into it.
This was a splendid presentation,
Thank
but you might help me. Pharmaceutical companies have marketers and scientists, and they're not equal.
Yeah.
The companies are dominated by the marketers, and they favour the treatment of chronic disease over, for example, I'm a physician, the development of antibiotics. So we're not developing any antibiotics. Why don't we consider antibiotics as orphan drugs? And do we need to revisit the whole definition of orphan drugs, because we know those things about pharmaceutical companies?
Well, yeah, that's a great question. Is it worth taking a few on notice? I mean, a few together rather than on notice, or
Anyone want to add to that question? We'll just start answering it.
okay,
and I'll
great.
keep an eye out for anyone.
Yeah, so that's a really good question. So yeah, I guess the TGA has its own sort of criteria for orphan drugs, which is about, as I mentioned, the sort of population available within Australia that could be the potential recipients. So what I understand of your question is, I guess, about understanding the potential for antibiotics to be more targeted therapies, essentially, that we could use precision medicine infrastructure to develop targeted antibiotics. That's not something my colleagues are working on. They are working, some of them are working on vaccines, so I could certainly direct you to them to talk about vaccines. But yeah, I mean, my sense is this precision medicine infrastructure, there's this kind of liberal discourse that Obama initiated this All of Us program as a way to reconcile all of the, horrors of, the Tuskegee experiments and all the kind of power asymmetries that existed through the biomedical complex in the US. And that this was a way to sort of say, look, can we put that aside and come together as All of Us, which is what that photo sort of showed. But a more cynical reading of that project, which is really about developing massive biobank information and a longitudinal database to go with it. So it's both kind of bioinformatics and telling the narratives of patients, would be to say that this is providing resources, virtually free of charge for pharma to then mine to find new drug targets, basically. And that one of the lines that we're working in the book is that this move from a discourse of individualized medicine to precision medicine is to sort of drop the pretense that this is actually about individuals at all, right? That this is actually about, finding the right subpopulations and matching them with the right drugs. And so the whole sort of orphan drug concept in that context becomes more about, pharmaceutical interests and profit maximization rather than about necessarily developing new drugs or about, as you say, developing a next generation of antibiotics. So, yeah, I don't have thoughts specifically on how we might lobby the TGA, but I'd love to talk about that.
Any other questions?
I was interested when you were talking about the proliferation of disease categories, particularly in relation to osteoporosis and the drugs that have been pushed to treat that or to prevent it. Or you've got new categories now of osteopenia, where you haven't quite got osteoporosis, but you could be on your way, therefore you should take this drug. When you consider that 2/3 of Australians over the age of 50 have low bone mass, it's bonanza for those same drug companies that were pushing HRT. And what it means for an older person, particularly an older woman, is that you're pressured and tested, and it gets to the point where you just want to avoid medical treatment, medical diagnosis, going to the doctor altogether.
Yeah, thanks. Yeah, I mean, this is really where people like Joe Dumit have done great work, and his work and his book, Drugs for Life, I think is the sort of definitive, critical account of that exact dynamic. That there's this kind of ambiguity now about what is considered to be a healthy or an unhealthy body, thanks largely to the kind of authority that is being propped up in the biomedical profession, thanks to, I guess, precision medicine is just the latest iteration of that. And I think what he shows beautifully in that book is the way that the kind of this ambiguity about what might be considered a treatable body is being pushed to in the interests of pharmaceutical companies to sell more drugs, basically, and that their ideal patient isn't actually isn't actually a cured patient, as the kind of economy of hope in the Biomedical Research Future Fund video seems to suggest, but it's actually a patient dependent on more and more drugs over time. So yeah, I think that's a very astute comment that you're making. I wasn't sort of aware of the osteo context specifically, but when you read through the sort of financial reports from the industry about where they see potential growth, I mean, it's sort of all there really, where they talk about promising drugs for these emerging categories of disease, and it's very much this dynamic that you're describing, right?
Hi, that was great. A couple of slides ago, you had a question there: is precision medicine producing facts? Just wondered if you could expand a bit on what you know, that bit of your presentation and what you mean by that, because I think that would be really enjoyable to hear more about that.
Thanks, Suzanne. Yeah, so part of my preparation for this talk, I've been reading some of your papers, and I really sort of got thinking about the way the infrastructure of precision medicine is being represented, and some of the ways that the crisis in the pharmaceutical value chain is being confronted by people, and really what's sort of being proposed in place of this broken blockbuster model is just a set of information flows, and it sort of moves. This sort of dream is that you now have basically data moving from the laboratory into various biorepositories, and where suitable, into regulatory settings and into clinical trials, and that the vision is really just about sort of speeding up the information flow across these domains. But what's really underdeveloped in all of these accounts is any sort of sense of power, is any sense of institutional authority, is any sense of who is actually producing worthwhile knowledge about how, essentially about how we actually come together and decide in each of these stages which is worth pursuing in terms of these massive numbers of compounds that could lead to therapies later on. So I guess what I've been sort of wrestling with in this sort of precision medicine space is how a more kind of STS account of fact making that accounts for the power asymmetries that we've just been talking about might actually operate. So that's sort of where we're going with the back end of the book. Yeah.
I have a bit of a sidebar question.
Yeah.
It's not your particular focus, I know, but really interested in that comment that you made about the slide from model to patient and the sort of human and non-human animal relationship, and what types of ethical issues, I guess maybe in a broad stroke, that you can think of, but also how that might affect that type of economy that you were talking about as well. And maybe even, thinking about the osteo sort of example and the types of patients that are expected of these subjects. Can the non-human animal become a dependent, a drug user, in this type of, pharmaceutical economy?
Yeah. That's just a massive question, right? Like, I guess that's that sort of opens out a whole set of questions about animal welfare that I've been trying to avoid in this in this research, but I think are incredibly important ones when you're considering like where a lot of these therapies are actually ending up, right? That that this is about managing cattle and managing kind of chickens that are being prepared for slaughter, basically, and these drugs are ending up in your body if you're eating, chicken, or often even if you're not, thanks to the emergence of this kind of feedback between drugs and environment through the water supply. So in terms of ethics and animal ethics. It's just an really an enormously complicated terrain, and I guess where I end up is more trying to emphasise the kind of cultural dynamics about which animals we're kind of happy to kill and which ones we're not, and that the dog is this kind of weird, is this kind of weird sort of space in the middle there between the smaller animals and the ones that aren't. And there's a guy, Brad Bowman at Harvard, who's doing a big research project on the beagle in the laboratory over the 20th century. So all the development of anti-smoking science was actually not people chugging down cigarettes and then figuring out who got cancer, but they were giving cigarettes to beagles. So it's a very complicated ethical terrain is the way I'd very inadequately answer that question.
Any other questions from the floor? Gary?
Thanks, Declan. It's really interesting work, and this is going to be a very half-baked thought, but I'm just wondering where practices of resistance might come into this, because when you present some of this stuff, sometimes it's so monolithic and institutional and big money and pharma companies and marketing. But this shift in kind of what's going on in funding and institutions and the practices of pharmaceutical companies and R&D, obviously because the practices are being remade, there's a possibility for the thing we call evidence and facts to be remade in other ways and medical subjects to be remade in other ways. And I suppose in thinking about things being remade in other ways, I'm wondering where you might see practices of resistance coming into any of these spaces.
Yeah, look, that's a great question, and it's one that I'm often confronted with in my research around climate change. Is like, this thing is so big and so hard, how do we do anything about it? So one of the ways that we're sort of confronting that problem of resistance and power dynamics is documenting the kind of economy of hope that is played out and imagined through young postdocs in these labs. Like, how are they motivated to turn up to these places every day and do what they do? I mean, it's a very kind of basic sociological question, but it's, I think, a really fundamental one in the context of what I've described, right? That there's real sort of disenchantment about the model that they're working with, about whether it is in crisis and how, that was the most sort of big picture version of it I could present. But within these laboratories, it's a very sort of nuanced way in which these limitations are being confronted all the time. So, I mean, one of the ways to think about alternative forms of power and resistance would be to look at the forms of care that exist in the laboratory, to look at the ways that they're trying to sort of challenge some of these dominant assumptions about who the imagined recipients of these drugs are, and that they're kind of opening up new spaces for new kinds of exchange to actually take place downstream. That's very vague, but yeah, I think it's a really great question that we've been kind of wrestling with in this research. Yeah.
Any further questions? I think I was sort of thinking about that in terms of the non-human animal kind of, if you shift the subjectivity attached to the animal, is that a potential point of remaking the subject and things as well? Sorry, who had their hand up?
I'm just a bit confused about the slide in terms of the meaning of control and discipline in both items 1 and 2. Control, who's controlling what? Control displacing discipline, I don't quite understand it. And the same thing with control parasitically depending upon discipline. Who's controlling what?
Yeah, so this isn't actually about the word control, but about the concept mobilised in a very particular paper that I talk about in much greater length. So yeah, that's an entirely inadequate summary up there. But basically, in Foucault's work, he talks about societies of discipline and societies of control as two kinds of forms in which he sees power operating and in which they're spatialised and temporalised in different ways. So he has this kind of diagram of operations of power, and there's a very live discussion in the kind of critical theory literature about how we might think about the relationship between these two forms of power in contemporary society. And I'm basically using these concepts as a way of thinking about these political dynamics, where societies of control is one way to think about this kind of diffusion of critical judgment across society, and disciplinary power is more about the sort of high modern institutions of schools and hospitals and the like. I know there are people who are much more versed in Foucault in the room than I am, so I'm probably going to stop there before I put my foot in my mouth too much further.
I think Foucault is always a good spot to stop on, so
yeah.
take this opportunity to thank Declan for the presentation today. Thank you very much.
Thank you.
And don't forget, it is his birthday, so if you would like to offer him personalised birthday wishes, I'm sure he'd be receptive to those.